get started. So one of the things I plan to do as part of this talk is actually to cover what we call as foundation models, and specifically for complex biological systems. And as I go along, I'll also highlight some aspects of design that we are trying to do with these types of models. And my hope is that I also want to get some feedback on what you all think about this particular approach. And we'll go about talking about them as we go along. So this project is actually something that was started as a BER pilot project, which was about engineering microorganisms and specifically about using CRISPR-Cas9 in terms of biocontaining them. So what normally happens is you have a lot of these engineered organisms that are out there, which can potentially get into the environment and be devastating. So if their dominating effect are actually like present, they might actually transmit their genes, and that can be a bad thing. And so there is a lot of concern about getting these systems working in such a way that you don't have to, you can protect against such events in the future. So our proposal actually was about building these micro communities of organisms in such a way that they could be secure. That means that you could essentially stand them up for a particular function, and they would stand down after their function is completed. So there are various ways in which you think about it. I'm not the biologist here, so I'm not going to explain all of this in very great detail. You can think of having a bunch of these Lego blocks that are assembled to form these microorganisms, eventually having this ability to do constitutive expression, which means that when an organism, two types of organisms are present, one can detect the presence of the other and perhaps inhibit its growth. The second one is about detection and response. So basically, one of the organisms will send out a signal which the other organism will sense and then basically respond. And the third one, which is more of a programmatic way of thinking about it, is a probabilistic response, where you have the signal that comes in. There are some things that act as producers. There are some things that act as a defender. So you can think of it as a sort of immune system, right? Very primitive immune system where you can understand how to protect yourself against a set of invaders in the community. So these were the scenarios that we sort of built up and said we will be able to do this by using machine learning techniques and AI approaches. There's a joint project between Argonne, Berkeley, JGI, Northwestern, and University of Chicago. And the core of that actually is about building this integrated platform for rapid engineering of biological parts. And in this case, it so happens that we think of the entire cell to be like a Lego building, and you have various pieces of the Lego that you need to assemble. And those parts of the Lego consist of transcription factors. You have enzymes, you have genetic parts, and all of these need to be built up in some sort of closed loop automated system so that you can rapidly test out what's happening inside of the cell or inside of the microbiome that you're interested in looking at. And all of this stuff that you see at the other side, all of this stuff that you see on the other side, which is this high throughput testing and so on, is done in sort of a liquid handling, cell-free system sort of an ability. So we don't have to do them in cells, express them, and do all those things. So the idea of doing this in high throughput is actually one aspect. And the second aspect, which also I'll highlight as we go along, is about this aspect of reproducibility, and more importantly on building the AI tools that let us do this in a proper manner. So if you just forget about the various parts that we need to build, at the core of this is idea that we have an AI-driven experimental design platform that lets us actually understand how to basically engineer any sort of biological construct that we need. So when it comes to biology, the first thing that we start thinking about is this hierarchy of information that's present. So what is this hierarchy? So if you look at proteins, they're made of amino acids. They have they have a primary structure, which is defined by their amino acid sequence. They have a secondary structure that is defined by their, interactions. And they have a tertiary structure, which kind of defines their third order, location and how they are interacting with each other. And that information is actually coming from central dogma, right? So a DNA, which is basically a nucleotide sequence, codes for that particular protein, and that protein performs certain function. So how do we think of this biological hierarchy and information explicitly? Or how do we think of it in terms of an implicit representation? So a lot of it has to deal with the fact that the current models for actually like learning representations of biological hierarchical information is kind of hard. And in this context, we'll introduce something called as the genome scale language models. Okay. The second part of my talk is going to focus on how to understand this biological information. So in the context of designing genes or proteins, and then looking at pathways. And if you're interested in pathways, I'd highly recommend you talk to Priyanka Shetty, who's presenting a poster today, where she'll talk about how we engineered a biological pathway using machine learning to kind of do threonine production, and then extend that to the genome scale. So I won't speak anything about the pathways today, but I'll just talk about individual proteins and then at the genome scale. So how do you extend these genome scale language models to understand how genomes are varying? And I'll present that evidence from SARS-CoV-2, which is a famous example. And the last one is, can we generate new examples that are also respecting the rules of what we understand with respect to these proteins and systems that we are trying to model? And this is the place where we start thinking of biophysics and we start thinking of experiments and simulations, especially how do you alter these programs, alter these AI tools in order to achieve certain amount of understanding of what the biophysics looks like. And then I'll talk a little bit about some of the work that we are doing in self-driving labs and so on, right? So first things, which is the biological information and hierarchy. So if you think of hierarchical information within omics data, you can sort of understand this from a very simple schematic, which is that you start out with a single gene, which has maybe about 1,000 base pairs. This is an average number. That's why I use the big O notation. So the big O notation is basically a representation to say, on average, this looks like this, or it's an asymptomatic, asymptotic measurement of how many base pairs might be there. Then you go into viral genomes, which is between 10 and 100 kilobase pairs. And then you have plasmids and pathways, which are, again, the same size. Then if you come to bacterial systems, they're about 1 to about 100 megabase pairs. And then you have eukaryotes, which are more than 100 base pairs, 100 megabase pairs. And then humans, who are about 3 gigabase pairs, right? So there is this sort of hierarchy of complexity in terms of how the information is organized within biological systems. And that stems from having what we call as genes or gene products, which starts with this open reading frame, which we all know about. I think most people would be aware of how this is actually like organized. And when you look at the open reading frame, it consists of these things like the promoter, start codon, and the stop codon, and what that protein should look like in terms of its nucleic acid representation. One thing to note, though, is that the nucleic acid representation is essentially there are 64 codons that code for the 20 amino acids that we know naturally, right? So there is inherent information that is actually much more coded in the nucleotide sequences than what is available in the protein sequence. So note this point, and I'll come to that a little later. And then you have collection of genes, which are either as contigs or open reading frames, which is all something that we know again. And then finally, you have entire genomes, which are now collections of these entire set of ORFs, or a bunch of genes that are tied together in some particular order. So if you read this entire thing, it sort of reads like it's like a book, right? So if you consider one organism, it's a book, and all these chapters are actually essentially genes that are present there. And there is a certain ordering of these genes that essentially leads you to have some functioning mechanism through which these are like emerging from. So the question is, can we use techniques there to help us achieve some things? So protein language models, which I'm sure you would have heard of, which is actually all the rage right now in terms of like protein engineering, have been really powerful in terms of adopting this idea from natural language onto protein structures. And the idea is basically that you have the primary amino acid sequences, you represent that in some sort of a way, and you basically understand what those alphabets are. And then once you find some mistakes in the structures, you can sort of model, issues with the folding pathways and so on, right? A second thing that you can also do is with respect to the secondary and tertiary structures, you can find out the relationships between the different areas of the protein. So for example, here is a domain switch that you see with respect to the N and C terminal. And it's basically some sort of a representation of saying, I'm happy when it rains versus when it rains, I'm happy. Both of them sort of convey the same meaning, but they have very different significance in terms of the protein structure. And you can extend that to even quaternary structure also. So that idea of using transformers, which are basically models that capture the sort of very complex relationships using protein language, has been there for a while. Of course, there are some challenges with respect to translating this natural language-based approaches for protein language, because in natural language, you actually have some ways to distinguish what is a word versus what is a phrase and so on. That doesn't exist with respect to protein sequences. They're a contiguous stream of, amino acid sequences. And then you have to make some modifications for the attention mechanisms, which is what people have done, and it's been done very well, actually. But you can think of equal number of problems here in terms of having the first transformer architecture that came out for protein embeddings, which was like Burkhard Rost and others who basically built this. And then you saw the AlphaFold revolution. And then there are like more of these new types of models that are coming out, ESM, TAPE, and ProTrans, and so on, which all have this idea of using the protein alphabets as their way to understand protein structure and doing design and engineering of these things. And of course, they have resulted in tremendous applications. So you actually have like lots and lots of examples. One from David Baker's group, where they designed an entire, complex, which is a very complex mechanically coupled axle rotor protein assemblies, as well as the fact that you could look at SARS-CoV-2 mini proteins that inhibit the, receptor binding domain. So unfortunately, though, the issue with this is that the implicit representation learning of this doesn't pay attention to what we understand from the biological hierarchy. I've been stressing about using nucleotide information and so on. And the idea that if you put in a sequence, it's going to give you a sequence out. But that also means that if you put a garbage token in, it's going to give you a garbage token out, right? So there is no notion of actually looking at evolution or any other bioinformatic inputs that we actually have a lot of access to, especially if you look at the genome level organization, right? And this is difficult to test if the latent space has any explicit meaning, because the models are essentially a black box, and there is this need for actually interpreting what these results mean in this context, right? So this is where we said, okay, let's start with the nucleotide sequence directly, right? So if you start with the raw, sensitive. If you start with the raw nucleotide sequences, then you have this advantage that you can use some implicit representation from these nucleotide sequences to go beyond what we call as traditional K-mer-like models. So in the K-mer length models and so on, you do have this issue of saying, what is the right length of the K-mer that I should be using? Right? That usually becomes a dependence in terms of how you end up coding your program and so on. And that can have issues with respect to the performance of the algorithm at any level. And at each level of this hierarchy, we also want to maintain the level from the previous token set, so we don't actually like lose out information. So when I actually move at the level of the enzyme or a particular protein gene sequence, I'm looking at the codon level tokenization, which lets me actually capture this information. And this is the place where I can use something called as generalized pre-trained transformer models, GPT models. And we'll see whether these actually work on individual open reading frames very shortly. And then when you have the collection of genomes or collection of genes that you want to look at, you still can use the codon level tokenization, but you need to do some more build up on the hierarchy so you can actually capture the ordering correctly. And then if you want to do it on entire genomes, this is going to be harder. And I'm not going to talk about this, even though we have some work in progress about this stuff, right? So one of the things is I'm not going to talk anything about how we built the GenSlim models. They're basically modified versions of these GPT-3 XL or GPT-3 Neo. And one of these algorithms that are standardly available for natural language processing, but adapted for the genome sequence. And we told us, let's learn what we learned from, 36,000 malate dehydrogenase sequences, okay? So these 36,000 malate dehydrogenase sequences are quite diverse. They come from various organisms. There is nothing that is like, special about them. They're all naturally occurring sequences. And we said, do we understand anything about that if we embed those sequences into this latent space that is represented by this GenSlim model? So these are genome scale language models. That's why they're called GenSlims. And within the context of the GenSlims, what you actually find is that if I stand at a particular point right there and move to one of the directions, so if I just move from here to here, you can see that there is a natural increase in terms of the GC content, right? I didn't give this as my input to the algorithm. I didn't ask the algorithm to actually go after this and predict the GC content. The latent space actually learns to represent the GC content in some sense of saying that this is one dimension in which you can learn how the sequences are organized. The second thing you can also do very simply is look at the nucleotide sequence length. So this is basically going from 850 to about 1064 or so, which corresponds to like roughly 300 residues, which is the natural length of the sequence that you observe. And if you move from bottom to the top, you can see that the length is actually increasing. Right? Again, this is completely unsupervised. The model basically learns it over the course of all the sequences that you present. And we have done a whole lot of experiments to kind of see how sensitive this is to like various other characteristic features, including molecular weight. And in some cases, we have access to experimental data that can give access, that can give insights into catalytic values or catalytic activity of the enzyme. And so we have looked at all those. And it seems that it is learning that representation. So there is some sort of clustering that's going on in the data. And these individual gene level models can now be used to interpolate and sample from this space, right? The argument is that when you take these samples and now say, give me a new example of this generative model, how does it look like? I can do that, right? I can get new synthetic sequences of MDH genes, and you can look at what they are actually looking like in the space. But before doing that, I want to make sure that I'm actually sampling something that is relevant. So we ended up building this huge phylogenetic tree of 36,000 sequences, and then trying to see where do these synthetic sequences fit in that part. So all the ones that you see here, the ones that are highlighted in black, and there are some that are on the top, you can't see them very clearly. I cannot fit all of them. And there are some also at the bottom, which are all synthetic sequences that are highlighted in black. And these sequences seem to fill in evolutionary gaps that we have not seen or sampled. And that is kind of interesting for us because we never expected them to actually start going after these sequences and trying to see if there are evolutionary relationships in these sequences. And this sort of helped us understand something, which is that the generative process that we were using was simple enough, but also sensitive to sort of fill in the gaps in the evolutionary tree that we had observed so far. So that let us do something, which is now I can feed these genes or the translated proteins into OpenFold or AlphaFold, whichever is your favorite tool. You can use one of them. OpenFold and AlphaFold are equivalent, so we're not going to talk about the differences. But what I want to highlight is that it actually learns the two distinct isoforms of MDH, which were present in the data. And we never gave this idea that it had two different forms of MDH or any of that data to the model. Okay, so if you go back and then look at this particular plot, what I want to highlight is that you see these little islands of places where you see some ridges. Like if you see, pay attention to this region or this region, there are some ridges which cross over, and there are some interesting locations from which we can sample the naturally occurring sequences. And when you end up doing that, you end up discovering that there are these two different isoforms of the enzyme. And in each of these cases, for some reason, we can actually see that the catalytic residues, as well as residues that are far off and implicated in the catalytic activity, are actually conserved. Right? So again, we started with the nucleotide sequences. Now we are using the translated protein sequences and then trying to model them using the OpenFold structure. And we do find the structures where they conserve the secondary features, secondary structural features. They also have these interactions, core interactions that we define are needed for the protein's function to be conserved. And it so happens that it also discovers a secondary form of MDH that we hadn't given any inputs on. We didn't even say what fold it belonged to. So now, one of my friends in the lab, who happens to be an expert in this particular protein, has ordered the sequences. We have them in the lab. They're testing it out for activity and characterizing it. So that's something that we are still working on. And this is not yet published or anything. So, please be aware of that. But again, the fact that we are able to stabilize them is kind of interesting. Now, I can do something even more, which is that I can start extending this to model entire genomes, right? So the idea was to say, okay, can I extend the GenSlims to understand the SARS-CoV-2 evolutionary dynamics? And the reason for using SARS-CoV-2 instead of a genome like E. coli is just the length requirement. So we are at 30 kilobase pairs, which is actually the right length. If you go anything more than that, any of the algorithms that you're using on GPUs will fail. So we had no other option but to like stick to something that was really small. Okay. So the idea behind using GenSlims was basically to see if we could test these genome scale models on a 30 kilobase pair genomes, which corresponds to roughly 10K amino acid sequences, right? And the hypothesis that we wanted to see was whether the GenSlims can capture implicit evolutionary information in the SARS-CoV-2 genome. So we wanted to identify which portions of the genome are evolving relatively fast. And we also identified what portions of the genome are coupled. So each of these are individual codons that are present in the genome. And the lines that you see are basically coupling that you observe in the evolutionary history of the data. This is all coming from looking at the attention mechanism and asking questions about what the attention mechanisms mean. So when you think of interpretability, the attention values are actually very meaningful in terms of looking at how they couple the different regions of the genome entirely. So in some sense, we wanted to prospectively model whether the SARS-CoV-2 evolution can identify certain variants of concern, and their modifications can be identified even prior to what was done using traditional approaches. One of the things I mentioned was that the length requirements are something that we really need to be worried about. So now you have a collection of genes, but that's not really very easy to translate. So you have a GPT-2-like model, which has 8 layers and 8 attention heads, which gives you a 10240 by 10240 positional encoding. So these are codons, so multiplied by 3. So you have the entire nucleotide sequence. And this model basically is trained on these ORFs, right, the protein coding regions. And then what you need to do is learn what is the relative ordering of the different sequences and how they are actually like put together. So there is a certain order in which the ORFs evolve, and you want to find out if this is actually possible. If it was a bacterial system and you had all these various types of reorganization going on, it's a very different story. But for these types of systems, it's actually quite cool. So we use something called the stable diffusion models. I won't talk about what these models really do, but the basic idea is that they read and learn the correct context of the ordering of the ORFs. Okay? So one way to think about it is now I'm going to build what is called as a foundation model, which means that I could train on all available COVID sequences and then be done with it, right? But that's not a good idea because the variation that you see in the data for COVID is fairly small. Right? Very small number of residues actually keep changing, or very small number of nucleotide changes are actually visible. So in order to make it more generalizable, we said we're going to train the foundation model on all available about 110 million sequences from what we call as the Patrick database. So it's basically the viral genome resource center from which we could download the data, right? And then we fine-tune the model on the SARS-CoV-2 ORFs. The idea is that once you do this foundation model, all the cost of computing only goes into post-training or fine-tuning the model. On a small number of these genomes, we don't have to run many times. And if you were to take the individual genomes and then say, I want to do this, we can periodically retrain. So let's say I get a stream of this data every week or so. I can do this a couple of times, and then I'm done. And then we're going to characterize the performance on different systems. I'll talk about that later. So you have the trained foundation models. You have the core scores that come out of it, which includes the semantic similarity, which when I showed you the latent space I can measure from, which is basically the distance metric that says how closely 2 sequences are related. So that comes from the embeddings directly. And then you also have the sequence log likelihood score, which basically says how good am I in predicting the next set of codons that I've observed, right? So the sequence log likelihood score gives you a value to basically measure fitness evaluation, and the semantic similarity score gives you the immune escape score. But this is not really biophysical in some sense. We are missing some details. So that's when we actually said, okay, let's use OpenFold. And OpenFold will take some of the regions of the protein that we know are important, for example, the spike and the RBD domains, spike RBD domain with the ACE2 receptor, which is actually giving you this fitness evaluation. So lesser the score in this case, it means that it is more fit towards the ACE2 binding. And then you have the epitope alteration score, which is basically a measure of saying, how does the spike RBD escape binding to certain types of antibodies, right? And we have a list of antibodies against which we can target, and that's what we do. And once you count the number of interactions that are present, you can measure the immune escape score, and those two are actually like put together to what we call as the variant of concern score. What is more interesting is the fact that you can take this idea even further. So now I have a trained foundation model. I can use this diffusion model that I was talking to you about to find the hierarchy of gene organization and generate new samples, right? And once I start generating these new samples, I have these synthetic SARS-CoV-2 genomes, and I can get into a lot of trouble for saying that, but I'm going to use that term here. It's a synthetic SARS-CoV-2 genome, which we can now measure the semantic similarity as well as the sequence log likelihood score, and do the same thing with OpenFold so that you can get the variant of concern. So you can sort of ask this question, can I prospectively measure what is changing in the SARS-CoV-2 genome that I might have not paid attention to in the wild? Okay. So does this model really work? Yes, it does. It does reveal the intrinsic patterns of how the SARS-CoV-2 is evolving. So we do the variant clustering. On the right-hand side, you can see alpha, beta, gamma, and all these different things that you observe. And you do see that there is clustering in the data. There are some sequences like Omicron, which have 32 mutations, so they are slightly more spread out than what you see. And of course, this starts spreading from the B level of the hierarchy, so that there's some implicit representation of the relationships between them. Okay. And not only that, we can also look at the distances in the latent space in terms of their GC content as well as the sequence length. And you do see that there is nice clustering of the data with respect to what we got from the entire genome, right? And one of the things that you might ask is the semantic score actually important? Does it do something important? You can start measuring the distance with respect to each of the variants that we have observed. For example, let's say, I'm not going to explain everything. Let's say I take the Omicron distance, and then I start measuring the distance with respect to these paints. What you observe is that most of the Omicron sequences are at the bottom. And if you go to the previous one, you can see that most of the bottom are actually Omicron sequences. And you do see the presence of these genome sequences with respect to the rest, which are like kind of farther away from it, which is a nice story to say that the semantic embedding actually has some physical meaning for interpreting how these sequences are farther off. So if I generate new sequences and then place them on the map of this phylogeny that we reconstructed, it's kind of interesting that the generative process also generates most of these sequences where we can see where the thing is evolving. So just to give you a sense of where the Omicron is, there are about 40 mutations that are present. And these are the mutations that it feels it can actually figure out into the future. And this is a very short window of evolution that we have run through right now. And there are some that are more than 80 mutations or so that are present in the data, but it also takes into account what is seen in terms of the evolution. And now we can prioritize them based on what these variants of concern scores are. You can look at the number of neighbors it actually finds. In many cases, it might not be very interesting. So you can see that these are all false. But in some cases, it does find there are interesting variants that you find. These are the ones that do have like interesting neighbors in the neighborhood. And so you can use that information to kind of figure out, you have new variants that you might have not seen, or even prepare yourself for seeing those variants in the future. Okay. There are some interesting aspects of this which comes to means, which is that there is scaling loss for these GenSlims, which we have to be aware of. So this represents some of the largest models trained on protein sequences or genome sequences. And in particular, if you look at ESM, which is one of the new models that came out, it is about 15 billion parameters, trainable parameters. Ours is about 25 billion. And that takes about 62 exaflops of computing, right? And then you can see the throughputs in terms of the batch sizes that you see across the different machines. And when we were doing this experiment, NVIDIA basically told us that we could use their supercomputer, which is 4,000 GPUs that are connected on 512 nodes. And we ran the experiments there to kind of see if there are any differences in terms of the sequence length. And we also saw that if you were to do the foundation model training versus all model training, which is on the entire sequence length of the SARS-CoV-2, there are significant differences that you see in terms of the throughput, right? And then we said, what if I want to train this model in less than a couple of hours? So that is when we subscribed to doing something on What is our ALCF testbed? It's basically a testbed that's combined with several systems, including Cerebrus, Samba Nova, GraphCore, and Grok. These are different architectures that do AI and machine learning hardware. This is openly available. Anybody is welcome to apply to accounts on them, and you can actually like use them for your machine learning jobs. And the Cerebrus CS2 chip happens to be a dedicated hardware. I'm not going to talk about what they're doing, but the idea is that we can actually train these models in a reasonable amount of time by clustering 4 of these CS2s together. In fact, this is one of the first applications where we have clustered 4 CS2 chips together to train the model in about 2.9 hours, which is a smaller version of the model. GPT-3XL takes about 5.2 hours. So which means that you can do it in a reasonable amount of time. Okay. Now I'll give a very top level overview of some of the biophysics. People have seen this many times in our group where we actually look at GenSlims and how we can integrate. So this is something that we did as an adaptive sampling technology for AI methods, how they can drive molecular dynamics simulations. In this case, let's say you're interested in running ensemble MD simulations. This case, we are showing the spike protein, and we are trying to understand whether there are some regions of the spike that open up. But unfortunately, that's very expensive to do it via single snapshot computing or other adaptive sampling techniques. So that's where we said, okay, we'll use our deep learning and artificial intelligence tools to build physically meaningful embeddings. That's the same idea that I was presenting to you about large language models. Use those to track the states that you're transsampling more often. And if you see that there are some interesting conformations, you actually like start the simulations from there. So the one that is shown with the tick mark, you start continuing simulations from. You stop the other ones that are not happening. So does this have practical value? In fact, it does. So we can actually fold proteins to about 1.2 angstroms resolution, which is, I mean, this is about 2 angstroms right now. This is actually the protein called BBA. And you can see what happens with and without machine learning being used in the workflow. With ML, we actually sample conformers that are close to like 1.81. That's kind of cool because you're now close to the native state of the protein. So you can sort of make advances in terms of understanding what they mean. But the important thing is that it actually gives you like 10 power 4x acceleration in terms of the sampling efficiency. So we compare this with Anton simulations and so on. I'm happy to like talk about it in much more detail. But this was also used in other simulations. For example, we use this for sampling the SARS-CoV-2 opening simulations with Rami Amaro's group and Lillian Chong, where we can sort of map out how the transition between the open and closed state happens in a relatively short time span of the overall simulation. So if they were to simulate this by hand, it would probably take them hundreds of microseconds of simulation time physically. But in our case, we could actually accelerate it to about several 100 nanoseconds, which is again an order of magnitude or 2 orders of magnitude speed up that you can see in terms of using these approaches. The second thing that we also did last year was how do we observe these very large conformational changes within cryo-EM maps, right? So we start with the cryo-EM data. We use something called as fluctuating finite element analysis, and then build all atom representations for them, which is a gradual transition from one state to another, so that we can actually model all of these transitions in a nice way. And there's a very complex workflow. This was another paper that was accepted as a finalist last year for the Gordon Bell submission. And in this case, we happen to use the experimental data as the starting point. We do custom meshing, which is the initial best guess. We run FFEA simulations, and the FFEA simulations are then passed on to this hierarchical AI, where the local fluctuations that are learned from the MD simulations are used to parameterize the meshes for continuum simulations. So we use all atom simulations that parameterize the meshes, and then using the meshes, we can learn these global fluctuations and then feed them back in terms of understanding whether we can refine the meshes even further. So how does that look like? This hierarchical AI approaches actually capture like 3 different levels. One is Anka AE, which is unharmonic conformational analysis enabled autoencoder. You're welcome to see the papers. They actually talk a lot about the methodological detail and so on. This follows the higher order statistical fluctuations in atomistic data, and then tries to build representations for them. Then we use variational autoencoders that kind of look at subunit level fluctuations. And the third one, which I think we are more interested in, which is this graph neural operator, which is actually a surrogate model for MD simulations completely. So they replace the MD kernels at shorter time scales so that you can actually like integrate steps at much faster time scales and then do like large fluctuation simulations. So this is work in reality. Here is a movie. I might not have time to like cover that, but here's the story where we started with this initial mesh. We let the mesh go and then look at the all atom simulations. This is a fairly large complex of the SARS-CoV-2 replication transcription complex. It's one of the largest complexes done on cryo-EM data. And it's an all atom MD simulation. And then we actually look at how to pull the RNA through the multiple binding sites. And as we pull through the binding sites, you can see it is like going through multiple conformational changes, which are not really visible when you just pay attention to all these cartoons that are moving. But when we come back to the mesh level details, we'll start seeing a lot more information of how that changes are actually undergone. So we parameterize the mesh using machine learning techniques. And those machine learning techniques are now learning the spring strengths that are needed between the different points on the mesh. So it's like an entire complex that's been modeled. And you can now start seeing what sort of interactions exist and why do we care about them, because that's actually kind of important for like looking at very complex structural changes that happen in this complex. And this is used to like refine cryo-EM data successively in an iterative fashion. So with that, the last part, I'm going to talk a little bit about how we are doing automated engineering. So this is actually about a system architecture for using these various types of robots that we have had in the lab. So this is a liquid handling robot. This is a much bigger liquid handling robot. There's a peeler, there is a sealer, there is an incubator. All these things have very different interfaces. But unfortunately, they lack a programming environment through which you can actually like run protocols, right? So what we have done is taken an engineering approach to do that. We have connected multiple opentrons, which are openly available robotic instruments for liquid handling, and then built an ecosystem of tools that can interoperate these robots together. And this is completely open source. You're welcome to try. Anybody having those liquid handling robots can actually play with them. It's all completely open. There's a whole bunch of people that work on it, including Priyanka, whose poster is there later in the afternoon. And we coordinate many of these different systems to like work on how we can do the automated engineering. There's a little movie here which kind of shows even simple things that we take for granted in the lab, where you're moving between the different instruments, like a human taking the sample and moving, can be very involved when it comes to a robotic instrument. Right. So here is this example where we have the Hudson robotic arm now trying to move the sample into a different robot, and then placing that in one of the opentrons. And it'll take a while for it to complete the play, but it's kind of meaningful to see how even very simple things that we take for granted inside a lab can be very painful for even simple maneuvers. But this requires a lot of planning and doing the AI portions to work correctly, because this is planning at the level of the robot and then trying to say, how do I get the robots to the right place, making sure that the coordinates are correctly visible. So we have cameras all over the place. There are sensors that kind of measure what is happening at any given time. And all of that actually comes together to, basically do what it's doing. They're kind of cool to see. It's unfortunately a 5-minute movie. I won't have like the time to like get everything. But there, it's going to move to the next portion where it is going to place it in the liquid handler, and then the pipetter will actually do its function. I'm not going to show that. And then we also have these abilities to build digital twins that experimentally we can reproduce. Again, this is built entirely and then deployed on a beamline. So there's a functioning beamline inside of APS where we built a robotic arm and tested it out and then put it back into the beamline so that we can run the experiments. And what we wanted to see was if this pendant drop experiment that's done automatically captures what we see in a hand-done experiment. It does, which is nice. So we are kind of happy about the fact that we can run these experiments. And then we're trying to like basically extract the methods out of these papers that exist for creating these protocols automatically. And Priyanka can give you a much better overview of what she's working on as part of her PhD thesis in this area. And then we can talk a little bit. So in summary, what we have done is built these GenSlim models, which can scale to large data sets. You have generative models that provide suggestions for experiments to be done. So we can do integration with fitness optimization tools for design workflows, as well as scaling on entire genomes that can face many challenges. And we have like AI-driven simulations that are now being integrated into these foundation models so that we can do some more interesting stuff. And the automated platforms that we are designing for experimental workflows are kind of like the highlight of everything that we're there. Of course, this is not possible just by my group. The whole bunch of people involved from NVIDIA, including Anima Anandkumar, who is at Caltech and NVIDIA, who helped us do much of the AI and machine learning. Cerebras, where we have like excellent collaborators that work with us with every idea that we give them. Of course, Oak Ridge and TAC were supportive of the computing. Some of the other collaborators that are involved across the world, and of course, my students, as well as the people that are involved in the various portions of the visualization, as well as all the stuff that we do. So computing time, TAC, Oak Ridge, NERSC, and Argonne, and of course, funding and everything. So if you have any questions, I'm happy to take them. So thank you.